You know that feeling, right? That ache of loneliness? But what if I told you it's not just a feeling? What if it's actually a biological signal? Something as deep and powerful as hunger itself? Well, today we're going to dive right into the neuroscience of your brain's social hunger. I mean, it's such a powerful sensation, right? That deep, gnawing ache for connection when you feel cut off from everyone. Have you ever stopped to think, why does it feel so physical? Almost exactly like being hungry or thirsty. Well, it turns out there's a really good biological reason for that. So this brings us to John Cacioppo, a total pioneer in social neuroscience. He gave us this game-changing way to look at loneliness. He basically said, hey, loneliness isn't just some bad feeling we should try to ignore. Nope. He argued it's a vital biological signal. Think about it. Just like hunger pangs scream, you need food, Cacioppo said loneliness is your brain's alarm system, shouting that a fundamental need, your need for connection, isn't being met. And this idea completely reframes loneliness. It means it's not some kind of character flaw or a personal failure. Not at all. It's an evolved survival tool, a signal that's literally designed to push us, to motivate us to get out there and fix our social connections. Okay, so if loneliness is a drive like hunger, then how does our brain actually make that happen? How does it create that powerful urge? Well, to get a handle on social hunger, we first have to look at how the brain creates the feeling of wanting for, well, anything, whether it's pizza, a pay raise, or a good friend. We're talking about the neuroscience of craving. So, at the heart of all this is what you could call the brain's motivation engine, the reward circuit. The fuel for this engine is a chemical you've probably heard of, dopamine. This whole network is what cooks up that intense feeling of, I want that, or I need that. So, when you're staring at a piece of chocolate cake and you feel that pull, yep, that's your dopamine system firing on all cylinders, driving you to go for it. Okay, so that leads us to the big million-dollar question, right? If loneliness really is a kind of social hunger, does it tap into this exact same dopamine-powered craving system to make us want to connect with people? To figure that out, scientists had to start somewhere, and they turned to animal models to begin piecing this puzzle together. Now, obviously, you can't just ask a mouse, hey buddy, you feeling lonely? It just doesn't work that way. So instead, scientists study something they can measure: objective social isolation. Basically, they just house a social animal, like a mouse, all by itself. This lets them see the direct, unfiltered effects of being alone on its brain and what it does. And what they found was pretty wild. Turns out, isolated mice will literally work harder. They'll press levers, they'll do whatever it takes, just to get a little bit of social time with another mouse. And neuroscientists actually pinpointed this social craving to a specific group of dopamine neurons. But here's the really fascinating part. They discovered that the brain circuit that handles the pain of being alone is totally separate from the circuit that processes the pleasure of being with others. It's like a two-part system. And get this, it shows you just how deep-seated and fast-acting this whole thing is. We're not talking about weeks of isolation. In mice, it only takes 24 hours. Just one day of being alone is enough to send their social craving circuit into overdrive. It is an immediate, powerful, biological gut reaction. So, okay, the animal evidence looks pretty clean, right? It lines up perfectly with this social hunger idea. Isolation triggers craving. Simple, case closed. Well, not so fast, because when we shift our focus over to humans, the story gets way, way more complicated. And the reason it gets so messy is that when researchers looked at chronically lonely people, they found a pattern of behavior that was completely different from the mice. In fact, it was almost the exact opposite of what the animal studies would have us believe. Yeah, instead of going out and seeking connection more, people who are chronically lonely often pull away. They actually show less motivation to be social. Their brains get rewired to be on high alert for any kind of negative social signal. And here's the kicker. When they feel rejected, it's not the dopamine craving circuits that fire up. No, the parts of their brain that light up are the same ones that process actual physical pain. It's like social rejection literally hurts. So this is the central mystery we're dealing with. On one hand, you have animal isolation, which clearly leads to more social craving. But on the other hand, you have human loneliness, which often leads to social avoidance and withdrawal. We've got two totally opposite reactions here, supposedly from the very same biological need. Which of course brings us to the question that's probably on your mind right now. What gives? Why is there such a massive difference between the animal data and the human data? Does this just blow the whole social hunger theory out of the water? Well, not necessarily. The answer isn't that the theory is wrong, it's just a lot more subtle than that. It all comes down to a really critical difference. It turns out the scientists studying animals and the scientists studying humans have been looking at two fundamentally different things. The research basically boils this whole contradiction down to three main reasons. First, they're studying different states, what it means to be lonely. Second, they're using totally different scientific methods. And third, well, there are some pretty key differences in the wiring of rodent brains versus our primate brains. And this first point is really the heart of it all. The mouse in the lab is objectively isolated. That's a physical state. It is literally physically alone in a cage. But the human studies are looking at subjective loneliness. That's the painful feeling of being disconnected, which, as we all know, you can feel even when you're in a room packed with people. And those are two completely different things. So what does this all mean? It means the social hunger theory is probably still right. Being suddenly and objectively isolated for a short time likely does trigger social craving in us, just like in the mice. But chronic, subjective loneliness, that's a whole different beast. It's way more complex psychologically. It gets tangled up with fear, with anxiety, and all of that can end up overriding that initial healthy drive to connect. It's like the alarm bell is still ringing, but it's become so loud and scary that instead of running toward help, you just freeze up. So, the big question for scientists is, how do we finally bridge this gap? Well, the research points to a pretty clear path forward. What we really need are experiments that can safely and ethically create short-term, objective isolation in human volunteers. Then, we can pop them in a brain scanner and see if their craving circuits light up, just like we predict. Another big step is to study animals with more complex social lives, think non-human primates, to get a better model that's somewhere between a mouse and a human. And, this isn't just some abstract scientific puzzle. This all leads to a really powerful, real-world question. If we can finally, truly map out the brain circuits for our social hunger, if we can understand both the craving that pulls us together and the fear that can push us apart, just imagine the possibilities. Could we one day develop brand new, more effective ways to help with conditions like social anxiety, depression, or even autism? You have to wonder. Maybe understanding this fundamental human need is the key to unlocking it all.